Good afternoon, everyone. My name's Duncan Richards. I'm Professor of Clinical Therapeutics here at the University of Oxford and a Fellow of St Hilda's College. I have been involved with a large number of other people in clinical trials looking for new treatments for COVID-19. And I just want to share with you some of the highlights and the lowlights of that experience to date. And there will be an opportunity to ask some questions at the end. I've got a brief presentation of about 20 minutes, just highlighting some of the issues and the challenges that we have been facing during the first phase of this pandemic, and I'm going to share those with you now. Just bring that up. So early in the pandemic, one of the striking features of COVID-19, and the official name for the virus is SARS-CoV-2, but the clinical syndrome that it causes is called COVID-19. Early in the course of this, there were some quite detailed descriptions of how the clinical course of this disease developed. And what this really showed was that the first week or so of people's illness was typical of an upper respiratory tract infection. But then around a week or so, in those that were going to do badly, there would be a remarkable deterioration in their condition. And this could be really quite rapid and lead rapidly to respiratory failure, requiring initially invasive ventilation, i.e. to go onto a breathing machine on an intensive care unit with all of the associated issues and problems associated with that. And unfortunately, a high proportion of those that required this ventilatory support continued to deteriorate and actually perished. A critical feature identified early in this disease course was that it was that those that had a poor prognosis had what was described as a hyperinflammatory picture associated with their disease. We all get a degree of inflammation associated with any infection that we experience. But it was a remarkable feature of this disease that you got a really dramatic hyperinflammatory response, and that's shown here on the graphs on the left-hand side. And you'll see in those that, with the red lines, that those that had a bad prognosis, there was this inexorable rise of these inflammatory markers across the board. And this led to an explosion of ideas about how we might address this hyperinflammatory picture in these patients as we move forward through the disease. And so as a result of this, in contrast to many of the previous respiratory illnesses that we have experienced, there was actually a huge range of potential therapies that were suggested and a huge range of clinical trials that were set up. Just to give you a little illustration of that, the first few publications around COVID-19 were really case series, and then, as we've seen on the previous slide, early biomarker descriptions. But then we moved very rapidly into interventional clinical studies looking at potential treatments. As you can see on the right-hand side, there was a dramatic increase in the number of those registered clinical studies over the course of the development of this pandemic. And as of last month, there were well over 2,000 trials, not 2,000 patients, 2,000 trials registered worldwide. And whilst this is an amazing response in terms of the number of trials and the amount of effort put in, there are some real problems here. Most of these trials are single country studies. And if you look across the European Union, of the 200 or so that were registered reasonably early in the process, about a quarter of them involved less than 50 patients, and so are likely to be generating only pilot types of information. The vast bulk of the studies, nearly 2/3, were trials involving 100 to 500 subjects. So a study of that size will give you some indication of the clinical utility of your intervention, but it won't be definitive. And it's actually only a very small proportion, less than a quarter, that are aimed at over 1,000 subjects that could actually give a definitive answer. We've heard a lot about hydroxychloroquine, and I'll mention a little bit more about that in a moment. But just as an illustration of that, there are currently over 140 clinical trials involving hydroxychloroquine. Now, there are multiple settings for the use of drugs in COVID-19, but it is inconceivable that there isn't going to be an enormous amount of overlap between those 140 studies. And it's a very real question of the utility of each of those when you have quite so many aimed at the same condition. So let's think about some of the things that have been done well during the response to this pandemic. And I want to highlight the recovery study. This is an Oxford University sponsored study, but is also the largest study in the world of COVID-19. This is the study that demonstrated the clinical usefulness of dexamethasone, which has resulted in that becoming now the standard of care for treatment of COVID-19. And it has also shown us that lopinavir-ritonavir combination and the hydroxychloroquine treatment are not associated with an improvement in terms of mortality. So how has this study been useful, and what are the key success factors associated with it? Well, it's what's called a platform study, which allows one to have a single platform on which you're going to test, i.e. you have common entry criteria for your study and you have common measurements that you're taking across your study. You can then plug in and take out different treatments and be testing them on the same basis. What the recovery study did extremely well was move very, very quickly at the beginning of this pandemic. It took a remarkable 9 days from the protocol being conceptualized on a bus in central London to the first patient being enrolled. That is something that commonly takes between 3 and 6 months to achieve. And so there was a real coming together of the research infrastructure in the UK, including the regulators, to make that happen. And as a result, by getting in early and coming up with a study that was needed, this is a protocol that has now enrolled over 12,000 subjects. It is also an extremely simple protocol. If you are trying to manage large numbers of sick patients in the pressured environment that we've seen in all of our hospitals during the first wave of the pandemic, you don't have the time for really complex and fiddly procedures. So what recovery did particularly well was have a very simple online protocol, which was accessible to all, simple support materials. There's no traditional case record form. In this case, all of the data is pulled directly from the NHS computer systems. And the final success factor here was the consistent support of the UK Chief Medical Officer and the whole of the NHS system to say that this was the protocol that they wanted every hospital to enroll into. And they have over 176 NHS hospitals now involved. So these key success factors have really allowed us in the UK to provide definitive information on the utility of some of those treatments. But hospital patients are only part of the picture. And within hospital, there are different groups. So the recovery trial, as I've described, is for all hospitalized patients, but there are specific treatments that are being looked at in the most unwell patients, those in the intensive care unit. And I'm personally involved in a study called Catalyst, which is looking at some novel, less well-tested treatments. And we are looking to enroll particularly high-risk patients. But not everybody requires admission to hospital, and Oxford has been involved in a number of other studies which are looking at patients who are not in hospital. There's a study called Atomic 2, which is aimed at patients who come to the emergency department because they're unwell, but it's deemed that they don't need to be admitted to hospital, and that's offering the potential for those patients to participate in research and to take part in a clinical trial. I'm also personally involved in setting up a study. We will all have heard on the news how badly affected the care home community has been in the first wave of the pandemic. And we're looking at how we can use the hospital at home network, which is a network that reaches out from acute trusts into community care homes to deliver high quality research and the opportunity for treatment for those in care homes. We also have those in general practice, and those are covered by a study called the Principal Study. And there's also a study looking at prophylaxis in healthcare workers, another group that have been particularly badly affected by this pandemic during the first wave. All of these studies, many of which are actually national platforms, are all sponsored and run from Oxford. And so Oxford is making a very substantial contribution across the board here in terms of the search for novel therapies for COVID-19. So some things have gone well. I think we've made substantial progress in terms of identifying novel treatments, but I want to highlight some of the ways in which things have not gone well. Whenever there is an emergency, there is a tendency for people to say, well, some information is better than none, and it's most important that we move extremely swiftly to just generate something. And this rather busy slides illustrates the raw data from a study of hydroxychloroquine that came out very early in the process and received a lot of media attention. But what you will notice from the yellow highlighting there is the top group, which is the control group in this case. Almost a very low proportion of those subjects actually had the test, which was the nasal swab looking for the coronavirus, on most of the occasions. Whereas most of the patients who received the active treatment did have a test on most of the occasions. And this is the sort of basic fundamental gap in terms of data quality that, frankly, if one had a graduate student who presented you with data that was as thin and incomplete as this, you would send them back to do it again. In this case, this study was rapidly published, it was rapidly picked up across media outlets and had a really quite substantial impact around the interest in in hydroxychloroquine. And it wasn't until the recovery study was able to come along with much more definitive data showing that actually treatment with hydroxychloroquine is not associated with a mortality benefit, that we're actually able to put that aside. So I want to highlight that just doing something and generating some sort of data is not always helpful in this setting. And you'll be aware that a couple of the leading medical journals have had to retract significant papers from their journals during the course of this first wave because the data were found to be unreliable. The other piece that I want to highlight is sometimes the flow of information is not provided in a way that is most helpful to those that are seeking to treat patients. So remdesivir is an antiviral treatment produced by Gilead in the US. And again, this has had quite a lot of media publicity. However, the first data to be generated on this and put into the public domain for this treatment was a compassionate use program. And this was published in probably the single most prestigious journal in the world, the New England Journal of Medicine, on the April the 10th. And this is 61 patients, completely uncontrolled data, essentially just a series of narratives of different people and making conclusions about whether those people got better or not. And without the use of any controls, it's impossible to tell whether the effect that you're seeing is due to the drug or whether these people were going to get better anyway. This was followed up just over 10 days later by a slightly larger study which compared two dosing regimens of remdesivir and showed that they both had a similar effect. But again, this was uncontrolled data. So A is the same as B, but you have no idea whether A or B is more useful than having nothing at all. So again, not very helpful. And if you think about when this was all taking place, this was at a time when we had no effective treatments and people were desperate for information. It wasn't until another 10 days later that we finally get some control data from the ACTT1 trial on 1,000 patients, so a decent number of patients, where we get some actually randomized placebo-controlled data. And that did indeed show that remdesivir was associated with some benefit, but the nature of that benefit was to shorten the illness. The jury is still out as to whether this treatment actually saves lives or not. So I just want to finish by highlighting my view on how we could do better as we think about the second wave of this disease, as we think forward to the winter months, where I think most people feel that it is inevitable that we will see an uptick in activity of this disease. And we think about future pandemics. Well, there are a few key principles, some of which have not been adhered to in this first wave. We need to think about the biological hypothesis and the alignment with the stage of disease. In simple terms, the replication of the virus tends to be a particular problem early in the disease. As I've mentioned before, you then get this hyperinflammatory syndrome, which is associated with many of the severe complications of the disease. But at that point, in the majority of patients, viral replication is much less of a component of the disease. So if you think about that in terms of a biological strategy, really we should be thinking about using antivirals early in the disease and anti-inflammatory treatments somewhat later in the disease. But if you look at many of the designs of early studies, many of the early antiviral studies were done in intensive care patients who already had very, very severe disease. And indeed, some of the anti-inflammatory treatments were aimed at those that really had very little evidence of a hyperinflammatory syndrome. I'm a clinical pharmacologist, and one of the things that I am always interested in is there a good rationale for the dose and the dosing regimen that is going to be used. in this disease. For many of these studies, we have seen people using the standard clinical dose, which has been optimized for other non-acute conditions. And in many cases, some of the antiviral doses that have been used have been woefully below those that are likely to be needed to impact the virus. We really need to think about the safety of our treatments, and is the safety profile of our treatment commensurate with our target patient group? If you're in an intensive care unit, the benefit risk equation is very, very different from someone who is mild to moderately unwell at home. And another key feature of this disease is you need to think very carefully about drug supply. A number of the early studies were conducted on highly experimental treatments, which were only in phase 1. And even if they had shown any benefit, really there was very little opportunity for those drugs to be made available to patients because the number of doses available was so low. So we do need to think about our strategy in terms of ensuring that we've got a good biological hypothesis, that we are using the right dose as best we can identify it, that we're going into the right patient population, and that we do have a secure drug supply such that if this is shown to be useful, we have a way forward. So a couple of things to think about for the future. I think it's very clear already that there isn't a magic bullet for this condition, and that shouldn't be a surprise because there are almost no diseases across the world for which there is a magic bullet. Most effective therapy these days is based on combination therapy, and if you think that there are a very large number of potential therapies available, the number of potential combinations is of course factorially higher than that. And so we need a really rational approach to those combination therapy if we're going to get any useful answers. One of the things that we do have as a gap at the moment is an integrated therapeutic strategy in what we would call phase 2, which is that learning phase of early development, where we're trying to understand whether a novel therapy, which hasn't really been tested before, has the really the biological effect to suggest that it might be useful if we took it forward into a larger phase 3 study, such as the treatments that are being tested in recovery. And actually the UK is in a really good position to do this type of work because we do have an integrated national health service, and as you have seen with the recovery trial, open 176 sites, you really can have a national response in terms of research. So I'm going to finish there. Thank you very much for your attention, and I'm going to go over to some questions. So I've got one here. Is there any information about the role of Ecmo? So Ecmo is extracorporeal membrane oxygenation. Essentially, it is a mechanical lung which does the job of gas exchange for you. And that is because your own lung becomes unable to exchange gas. And one of the features of those that did badly with this condition is that their oxygen requirements, so the amount of oxygen you need to keep the blood oxygenated, went up and up and up. And of course, once you're on pure oxygen, there's nowhere higher than you can go, even if you're on a ventilator. And in a small number of cases, then you can use Ecmo to do that job of gas exchange for them. It is a bit like going onto a bypass machine. It's extremely complicated. It is associated with real risks for the patient. And the simple truth is we only have a small number of these machines available in the UK. And so it can be life-saving in a small number of patients, but the number of patients that we can offer it to is small, and they have to have a pretty good underlying health status in order to be able to benefit from it. I understand that the Oxford study is a follow-on from a report published from researchers in Spain in February. I'm not, so it is certainly true that, as I've highlighted, with 2,000 studies going on at any given time, it is likely that you will have multiple studies looking at the same drug. What you have to ask yourself is the quality of those studies and the question that they're answering. So Recovery is a study that is all about answering in essentially a very simple question, which is this treatment, which I'm going to offer very broadly to a hospitalized population of patients with COVID-19, is that treatment associated with a reduced progression to the intensive care unit or of death? And that is the question that the Recovery study is asking. And at a simple level, at a public health level, that is a question, that is the really most pressing question that we have to answer. We, like everybody in the world, have limited capacity in the intensive care unit. And you will have seen the heroic efforts that were made to create additional intensive care space to stop that being overwhelmed. So any drug that prevents progression to the need for intensive care and all of the resources that are required for that is something that is useful. And clearly we are in the business of stopping people of dying from this condition, and so that is useful. Other studies may address questions of whether your drug is addressing a piece of biology that is interesting, but these tend to be phase 2 studies. And the majority, as I illustrated early in the talk, the majority of studies that have been set up for COVID-19 are very much aimed on that early end to try and get sort of pilot data. But I would question the utility of 50 pilot studies when in fact there is the opportunity to do one definitive study if you move quickly and you do it in the right way. The number of international and mortality endpoint studies that have been conducted is very, very small indeed, whereas the number of pilot studies is much, much longer. So there's a good question here about how representative is the population that get into these studies of the general population. And it is a common feature of all of clinical research, and this is widely published, that it tends to be those of higher socioeconomic status, and those from the BAME community are particularly underrepresented in these in clinical research generally. One thing I would say about the response to COVID-19 was that the simplicity of the recovery study really enabled a very broad population to be enrolled into that study. It had a very simple consent process, very simple information that was rapidly translated into multiple different languages. And importantly, this was not a study run in the large, famous teaching hospitals, which are the usual homes of clinical research, but it's 176 sites, most of which are district general hospitals serving their local population. And whilst I haven't seen the detailed breakdown, although it will be coming in the publication, I expect that the coverage of the population for studies like recovery is somewhat better than it would be for other more involved research, I suspect the phase 2 studies will look much more like our traditional pattern, i.e. it tends to be middle class and white people who tend to enroll in those studies. And that's something I think a key learning from this experience is, how do we have constructs for clinical research that really allow us to enroll the full population, not just those that perhaps reside in North Oxford. So how do you deal with the interface between those colleagues who are on the front loads and those that are reviewing the large number of studies across the world? So one of the things that we did in Oxford quite early in the process was set up a working group that met twice a week. And that was made up equally of the people who were seeing the patients on the front line and those who were, like myself, incarcerated at home, but had free access to the internet and to the literature. And we found that this was a particularly effective way of ensuring that those on the front line understood and were aware of the very latest information emerging from the literature, but also that those that were thinking about and planning clinical research were aware of what it was really like on the front line treating these patients. I reviewed a large number of proposals from different groups who really had not paid any attention to the environment of the front line. And they had written very, very, what I would call traditional clinical studies, complex clinical studies requiring lots of blood tests and very precise time points and all of these sorts of things that we usually insist upon, but are totally impractical in the kind of environment that people were dealing with in the first wave of the environment. So we've got a question of, can the cytokine storm seen in the patients who become very unwell be prevented by aggressive treatment with anti-TNF alpha drugs? Well, I'm probably not the person to give you a completely unbiased answer to this, in that I'm one of those that have been advocating the investigation of anti-TNF treatment. It's worth saying a word on cytokine storm. This was a phrase that was particularly prevalent early in the disease, and really was used as a shorthand for this hyperinflammatory syndrome. What we've understood subsequently is that actually the situation is somewhat more complex. And what you get is, yes, there certainly is an upregulation of multiple cytokines, which are these signaling molecules that tell you about, tell the immune system to get on with it. But there are also big shifts in the nature and type of cells that are present, particularly in the lungs. And we suspect that actually it is the shift of some of those cell groups that are actually responsible for much of the damage and the poor gas exchange that we see in those that have a poor prognosis. And so what we have been looking at is, what is the best treatment for that? A number have been suggested, and dexamethasone clearly is effective and reduces mortality in this condition, particularly in those with the most severe disease. Dexamethasone, like any steroid, is very much a blunderbuss approach here. And whilst short treatment courses with steroids aren't associated with some of the long-term problems that we're all aware of relating to bones and skin and those sorts of things. However, there are some quite important neuropsychiatric adverse effects of steroids, which can be problematic, particularly in someone who has got delirium because they're septic from a viral illness, or are on an intensive care unit, which is a deeply unnatural environment to find yourself in. And so there are many of us who think that whilst dexamethasone is extremely important and must form a vital component of the treatment paradigm here, we need to continue to strive to look for other therapies. And one critical question that we're trying to address at the moment is, should we try to reduce a number of cytokines to a moderate degree, or should we knock out one particular cytokine completely? There are a number of drugs available that can specifically take out anti-TNF, IL-1 and IL-6. And there are clinical trials ongoing trying to answer that question about whether any or all of these are useful on top of dexamethasone in this condition. I personally believe that there will be utility from drugs such as anti-TNF, particularly earlier in the inflammatory cascade, because anti-TNF seems to play have a particularly important role, acting as a master switch of the disease. So there's a question there about what the therapeutic target should be. Should we be looking to cure the disease? Of course, we should be looking to cure the disease, and in reality, that will depend on the availability of a vaccine. There are enormous efforts, and Oxford is leading the way in terms of identifying a vaccine. Certainly, the preliminary data look like the Oxford candidate vaccine is well tolerated. What we don't know at this stage is whether it really protects people from infection or not, and we eagerly await those data. But there are some really quite concerning data emerging that the immunity that you naturally generate having had COVID-19 may in fact be fairly short-lived, and therefore even if you've had the disease, you may be at risk of a second infection if you're exposed to the virus again some months later. This is clearly something that we need to keep an eye on. And so in terms of a therapeutic strategy, yes, the vaccines are incredibly important, but as we've already seen, there are no vaccines available yet, and we are beyond the first wave in many parts of the world, particularly Asia and Europe, not necessarily the same in the US, but the fact is there is no vaccine available. And therefore, you also will need treatments that are available to mitigate the disease. We have two of those at the moment, remdesivir, an antiviral treatment, and dexamethasone, an anti-inflammatory treatment. But they by no means are the whole answer to the question. And I think it's important to say that whilst antibiotics for bacterial infections can be expected to effect a cure on someone who has an infection, in most cases, antiviral treatments simply reduce or dampen down the amount of viral replication. Most of the time, they do not knock out the virus completely. Even in controlled conditions such as HIV now, you can knock down that virus to very, very low and indeed undetectable levels. But the number of people who are actually cured from HIV from heart therapy, I think can be counted on the fingers of one hand. So there's a question here of, would it be, if you had a mild version of COVID and you got reinfected, would it be a mild illness? You would hope so, because you would hope that you have generated both an antibody and a T cell response, which would mean that if you are infected again, that you would get a mild disease. And indeed, this is in some ways the principle of vaccination itself. But I just highlight the fact that the evidence that is emerging is that might be quite that duration of protection might be quite short. and maybe incomplete. And this is the important thing, is 80% or so of people who get this disease do have a mild illness, but because so many people have been infected, you know, your risk of having a bad outcome is small on an individual basis. But when you multiply that up to populations, you know, in the UK, we've had 45,000 deaths, and worldwide, it's hundreds of thousands of people have perished from this illness. And so that is the problem. Is, yes, you may be lucky, you may be the one who does get the mild illness, and you may be protected, but it's far from guaranteed. And we have seen, you know, so many people be affected that there are, you know, tragic stories all over the world of, you know, previously fit and healthy people who have been laid low and indeed perished from this this disease. I've got a question about how the Oxford Recovery Study compares with other efforts across the world. So if you look at what was different about recovery, it is a single country study, it's a UK study, but it was across the whole UK, and it asked a very simple set of questions and had a very focused response to that. If you look more generally across the world, there is a WHO study called, oh, I've forgotten the name of it now. It's got a sort of balance type name, and which is also looking across multiple countries. But actually, recruitment into that WHO study has been slow. It was much slower to get started than the recovery study, and actually, the number of people it has recruited has been much smaller. There are very, very few other studies which are really looking at mortality in these studies. Most of the studies are using what is called an ordinal scale, which is an ordinal scale which rates people from essentially not having anything wrong with them, which is a 1, to being dead, which is a 9, with different scales in between in terms of disability or requirement for medical intervention. And what people are using that scale to do is to identify whether your treatment is able to move you up and down that scale. Now, that is useful, but you need quite large numbers in order to be able to detect those differences. And there are problems in terms of using that type of scale, because clearly it makes a very, very big difference if you move from number 8, which is being critically ill on an intensive care unit, to 9, which is being dead. However, moving from 3, which is you know, not being too bad outpatient, to being slightly worse as an outpatient isn't a great deal of difference. So there are problems with using those scales as a means to really translate the usefulness of your drug into clinical practice. So to what extent do we really understand why the few identified drugs have been useful? Well, if you look at, again, if we just use the example of recovery, they took on a mixed group of different treatments. So there was lopinavir and ritonavir, which is essentially an antiviral approach. There's hydroxychloroquine, which is also an antiviral approach. But you then have treatments like azithromycin and dexamethasone, which are essentially anti-inflammatory treatments. So within that study, there were different kinds of therapy that were useful. If you sat back for a moment and thought about what sorts of treatments would be useful for this condition, then it is, I think, clear that something that would reduce replication of virus might well be useful, particularly early. And then there would be a use for an anti-inflammatory treatment, which would be particularly useful later in the disease. Now, within that, of course, there is enormous complexity around exactly what antiviral and how you use it, and particularly on the anti-inflammatory front, exactly what kind of anti-inflammatory to use and the mechanism of it. There is a rationale for each of these treatments, but I would not say that we fully understand the whether that mechanism is useful in this condition. And I just highlight one particular thing is people measure the concentrations of cytokines in the blood. And what that shows is that those cytokines are elevated. But we already knew before this pandemic that actually measuring the concentrations in the blood is relatively uninformative in terms of clinical prognosis. It is the concentration and the effect of these cytokines within the tissue, which is what determines how well or badly patients do. And therefore, looking in the blood tells you that there is a fire burning somewhere, but it doesn't tell you exactly in which room the fire is burning, and it doesn't tell you whether all of the smoke that's coming out from that fire, what is really is that was causing the fire in the first place. And so just measuring cytokines does not tell you that it's going to be useful. And I just use a specific example here. If you look at the concentrations of IL-6, they are particularly raised in this condition. And there have been a whole series of clinical studies looking at various blockers of anti-IL-6. And for every positive study, there is a negative study. So we really don't know whether anti-IL-6 therapy is really useful for this condition or not. But you would think if you were going on the concentrations in the blood alone, that this would be incredibly important and would make a massive difference to patients. The truth is, it probably is helpful for some groups of patients and not others, but we really don't know who those groups of patients are. In the recovery, do you have any safeguards against medical nationalism, i.e. preventing the repetition with the remdesivir situation? So the treatments that they took on within recovery, they took a pragmatic approach. And if you look at most of those drugs, they are all relatively old drugs that are widely available, because one of the critical factors that they did take into account with recovery was the fact that if we show that this drug is useful, then we do need to have a clear path where the drug could then be made available to larger numbers of patients. And clearly, if you use a phase 1 drug where there are 14 grams of the stuff available in the whole world, then that's not going to be possible. I have spoken to the investigators of recovery, and the truth is that part of their decision-making process was pragmatic around which companies were prepared to make the drug available at scale to them, and not every company was prepared to do that. So there are some treatments that could have gone into recovery that did not go into recovery because the manufacturers of those treatments were not prepared to make them available without any strings attached in that way. And I think it is a really challenging aspect of this, which is to think about how we use the resources that we have available for clinical research to generate answers that are then applicable and usable by large numbers of people across the world. It is one of the reasons why I'm particularly interested in anti-TNF as a cytokine blocker, because there are about 6 or 8 manufacturers of anti-TNF products across the world. There are so-called generic manufacturers, biosimilar manufacturers now of these products. And so if we are able to demonstrate that this drug is useful, we are not limiting then the supply to a single manufacturer and holding everybody hostage to that. The truth is, the earlier you go in the drug development process, the more limited the choice you will have of manufacturers, because there just won't be very many. And so if you identify a really effective treatment that only has one manufacturer, such as remdesivir, then you are going to have this problem of scale. It's worth saying that remdesivir is particularly difficult to make. It's a real headache. It's a 20-step process, and it's a real headache to make remdesivir. And if you think about where remdesivir came from, it was being trialed for Ebola in Africa, and whilst that disease has been a scourge in certain parts of Africa, the scale and the numbers involved in that is a mere fraction of those of COVID-19. And so none of the manufacturing infrastructure was geared up to be on a sort of massive global scale. So why do young people apparently have a more mild disease than older people? The answer at this stage is we don't know. But the piece I would caution people about is that what we have seen is that if you're older, particularly if you're over 70 or 80, then you have a particularly high risk of having a poor outcome. And whilst that risk is much lower if you are younger, because of the number of people who have been infected, there are still a large number of tragic stories of, relatively young people, and indeed some very young people who have been laid low by this disease. The basis for that difference, we really don't know at the moment. There are some interesting hypotheses around the way that the immune system becomes less effective with age. We know, for example, that vaccinations are much less effective in older people than they are in younger people. And it is quite possible that in effect, if you are older, you get more of this unregulated inflammation and a sort of maladaptive response to the virus. Whereas if you are younger, you get an adaptive response. But this is frankly speculation at this stage. And the short answer is we don't know. But it is also important to remember that whilst if you are younger, you are at lower risk, but it is far from zero risk. We all need to play our part in protecting the whole population. There's a final question here, just in terms of, is it possible to treat therapies of the different kind of COVID-19 strains the same? At the moment, the antiviral therapies that we have are aimed at particular enzymes that are involved in constructing the virus, and they are quite general, and we have not identified particularly resistant strains for those at the moment. The the anti-inflammatory treatments are really aimed at the host, so the person response to the condition, not the virus per se. And so we would expect those to continue to be broadly applicable. But just to come back to your previous question around, the difference between young and old, it is very likely that different people will mount different kinds of inflammatory responses to this condition. And therefore, it may well be, as we understand it better, that we are better able to refine, particularly the anti-inflammatory therapy that we are able to offer to different groups of patients. But I have to say, we are still at the one-size-fits-all stage of therapeutics for this condition. So, thank you all for lots of great questions, and thank you for your attention. And I hope you enjoy the rest of the week of talks and events from St Hilda's. But with that, I'll sign off. Thank you very much.